Welcome, everyone. It's my pleasure to have you on board for this special session on optical genome mapping in cell and gene therapy. I'd like to give you an introduction on how optical genome mapping is revolutionizing quality control for the genomic integrity of cell products. Like research and biopharma communities have made tremendous progress utilizing cell and gene editing tools for therapeutic applications. The market for genomic medicine is booming with thousands of players in active trials, 60% of them addressing cell-based therapies with a distinct focus on oncology. Yet so far, phase 2 approvals are extremely modest, only 14%, reflected in just 28 products approved by the FDA. So part of this low success rate has to do with the challenges of manipulating cell lines in an in vitro environment. Put into some in vitro artificial conditions, cells are reacting and adapting to their new environment by undergoing tremendous genomic and proteomics changes. Hence, what may be once a cell colony with an homogeneous genomic population often ends up with an heterogeneous mixture of subclonal population responding to stress with mutations, chromosome abnormalities, and DNA damage. In gene editing, while maintaining optimal culture conditions, cells are even more subjected to DNA damage due to the incurred DNA breaks caused by random genetic insertions, adverse on-target editions, and off-target editions. So for quality control, it is crucial to survey and monitor these genomic changes with high resolution for the entire genome to ensure quality assurance and functionality in any downstream applications, especially for human deliveries. So these applications are overall summarized into two workflows: an ex vivo workflow producing cell products engineered for their functionality with or without gene editions and delivered back to a recipient as an autologous or allogeneic infusion with an eventual banking of the product as well. In vivo gene therapies are intended to deliver a genetic cargo, drug, or a molecule to recipients, which will alter the recipient's genomes within their body. In all workflows, a critical step of cell characterization is needed to monitor structural changes and avoid adverse outcomes. So what are these genomic variants? From this published metadata, and that is based on about 100 references, genomic abnormalities in HPLCs were identified across the entire genome with more than 700 recurrent non-polymorphic genetic variants in a population of merely 1,000 samples. The top plot shows a genome-wide recurrent copy number changes with prevalence for chromosome 12, chromosome 17, and chromosome 20. The bottom plot indicates that these variants are ranging from sequence-based mutations to anomalies. Moreover, structural variants are impacting the integrity of the whole genome with occurrences of balanced translocation and unbalanced translocations. So the take-home message is, put into culture and induced for reprogramming, cells are reacting with frequent structural rearrangements that can hardly be denied during cell characterization. In genome editing, there is an added risk of genotoxicity caused by off-target edits, usually the side effects of programmable nucleases. For example, the use of CRISPR-Cas9 editing tool generates double-stranded DNA breaks, which are repaired by either error-prone or fidelity pathways with or without genetic insertions. And when insertion occurs, as editing tools are tolerant for sequence mismatch, there is an elevated risk of off-target edits that must be evaluated carefully before any clinical application. In this publication, this is an example of an iPS-C production, but were edited for knocking out two genes, HLA-A and HLA-B on chromosome 6, to produce hypoimmunogenic cells intended for generic or allogeneic use. So it's not unusual to multiplex gene edition, and that is mainly to prevent longer culture periods and clonal selection, and therefore to avoid spontaneous mutagenesis. But multiplexing gene edits means facing a higher potential of off-target edits, as displayed in this bar chart. And when quality controls for cell characterization is poorly designed, catastrophic outcomes may occur. So on the left-hand side, a report of a critical cancer development in recipients for CAR T-cell therapy leading to one fatality. On the right-hand side, a report of improper cell characterization for an allogeneic cell line for cancer treatment that ended up not being what the developers thought it should be, causing a tremendous loss in investment and the termination of the program. So these dramatic outcomes, of course, triggered the scrutiny of regulatory bodies. So here are two examples of biotech put on hold by the FDA for a lack of rigorous product characterization before or after human delivery, or involving the detection of chromosome abnormalities, either during production, and that is the case of the Beam Therapeutics case, or during treatment, and that is the case for allogene therapeutics. So when it comes to product characterization, let's look at a typical workflow and in ex vivo production of cell and gene therapeutics. From cell collection to cell isolation and selection, to cell genomic editing and engineering or functional reprogramming, to clonal selection and expansion, cell characterization, back to cell infusion, there are some critical steps for quality control that are addressed by optical genome mapping, which I have mapped in this slide. So cell lines are selected for their ability to be functional, relatively homogeneous or pure, with the capacity to divide without being toxic or tumorigenic, and while maintaining cell fitness, that is to avoid cell death. So what are the tools needed to characterize cell lines? They are historically divided into two classes of methodologies, largely triggered by the size of the genomic event. Cytogenomics for structural variants to gene fusions, and then targeted sequencing for gene fusion, epigenetics, and sequence mutation. For cytogenomic testing, multiple techniques needs to be used to comprehensively characterize cell lines. Large structural variants with conventional karyotyping, to high-resolution CNV and allelic imbalance variants with SNP arrays, to absolute quantitation on few targets with droplet digital PCR. But what if we could adopt a genome-wide methodology with a detection range from structural variants to gene fusion, sensitive for clonal heterogeneity and cost-effective? So here come optical genome mapping, or OGM for short, a next-generation cytogenomic technique that can detect structural variants and gene fusions in a single assay at a higher resolution than conventional cytogenetics in a very cost-effective workflow. Now, note that OGM and karyotyping are the only two techniques for direct visualization of the DNA. All of the other techniques using DNA as a template to visualize genomic proxies. So let's look at how optical genome mapping is redefining cell genetic characterization. And I want to give you an overview of a very simple workflow for this technique. It starts with prepping the DNA, extracting ultra high molecular weight DNA molecules, which are going to be put into an electrophoretic field through the Bionano analyzer, and then image every single DNA molecule being imaged and then remapped to a reference genome. The interpretation occurs at the end of the process with visualization of structural variants that are exclusively into the test sample as compared to a reference. The comparison of structural variants is as simple as looking at the difference in the pattern of the test molecule as compared to the reference molecule. So here's an example on a live visualization on these molecules passing through the electrophoretic field into the Bionano analyzer. And the resolution is tremendously higher than karyotyping. Because of this, we can actually consider switching from a DNA contrast staining to DNA fluorescence staining, with the traditional conventional cytoband becoming molecular bands. And that is all of this viewed under a high-powered microscope. So technically, with a traditional karyotype, you may add about 550 bands. With OGM, it's about at least 500,000 bands in the form of fluorescent labels, increasing the resolution for abnormalities by 10,000-fold. So not only you can now consider genome-wide visualization of all of these structural variants ranging from deletions, duplications, inversion, translocation, tandem duplications, repeat expansion, as well as copy number and allelic imbalances. On the right-hand side of this cartoon, I'm showing you the differences in the pattern from the reference sample, which is in green, as compared to the test sample in blue, showing you where the software identified changes in the pattern, which are eventually interpreted into specific copy number changes. So in this example, gradual anosomy detection in the TUBA1 GFP cell line is increased with cell passages. As you can see on the top plot, optical genome mapping has been used in order to map these aberrations with extreme precision and with low allele variant frequency detection. From passage 20 to passage 40, for instance, you can clearly see the evolution of that cell line towards anosomies. But it's not only that. We can actually detect with OGM much smaller events. And the bottom plot actually shows just that. Deletions, insertion, duplication, and inversion at every single step of the production of this cell line. So that is for production of stem cell. How is it with the edition of the genome? And here's an example of the evaluation of optical genome mapping for on-target as well as off-target genome integrity using a particular cell line, which is meant to be modified and address the X-linked combined immunodeficiency disease. So on the left-hand side is a cartoon showing all of the different aspects of this edition, with a Caspase-9 editing tool cutting the genomic material. We expect double-stranded breaks or DNA double-stranded breaks, which are going to be rearranged and repaired with or without the edits. The preferred option is the middle one, with the expected homologous directed insertion of the genetic material, in this case, CD40 LG gene. However, in this type of analysis, there are some adverse reactions, namely abnormal structural arrangements or deletions that may occur proximal to the target site or outside of the target site. So the idea is to use optical genome mapping to map these genome-wide aberrations and also map the clonal heterogeneity associated with these aberrations. And this study has evaluated optical genome mapping with a detection of as low as 2.5% of variant allele frequency, which is far better than the limit of detection provided by the standard technique of karyotyping. So now let's hear it from experts in the field with or with a specific use of optical genome mapping technique, or with the goal of delivering reliable bioproduct to the market. Thank you for your attention.